Good afternoon. Myself Dr. Shashikant Swankar from CCM GMC Kachandur Durg. Today we will discuss upon microbiology. Microbiology is the biology of microscopic organism. Microbiology should be an easy word to define the science of a small life or the study of living things so small that cannot be seen with the naked eye. We can classify microbiology into pure microbiology and applied microbiology. Pure microbiology includes bacteriology for the study of bacteria, mycology for the study of fungi, phycology for the study of alga, immunology for the study of immune system, virology for the study of viruses. Microbial physiology is the study of microbial cells function. It includes the study of microbial growth, metabolism and the cell function. Microbial cytology is the study of microscopic and submicroscopic details of organism. Microbial genetics is the study of organism and regulation of gene in microbes in relation to their cellular function. For the applied microbiology, veterinary microbiology and pharmaceutical microbiology. Veterinary microbiology is the study of microbes of veterinary importance such as probiotics and pathologies which are related to the veterinary medicines. And pharmaceutical microbiology includes the study of microorganisms which are related to the production of antibiotics, enzymes, vitamins and other pharmaceutical products and those microbes which are pharmaceutical contamination and spoilage. Microbial biotechnology is the manipulation of microorganisms at genetic and molecular level to generate useful products like insulin from E. coli. Food microbiology is the study of microorganisms causing food spoilage and food borne illness. It is all the use of microbes to produce food. Microbiology is the study of microorganisms usually less than 1 mm in diameter, which requires some forms of magnification to be seen clearly. Compound microscope, initially it was used for microbiological studies. Basic parts we can see in this slide. This is the microbiology which we are used in previously. This is modern microscope including eyepiece, base, condenser, iris diaphragm, objective lens, body tube, mechanical stages and adjustment knobs. Why we need to the study of microbiology? Because microbiology are related to all lives in all environments, many beneficial aspects related to life process. Only a minority are pathogenic. Most of our problems are caused by microbes. Importance of microbiology includes medicinal uses, environmental science, food and drink products, fundamental research and agricultures, pharmaceutical industries, genetic engineering, recycling sewage, biomedical use of microbes to remove toxins, use of microbes to control crop pests. Timelines of microbiology. In this slide we can see the milestones of microbiology in the following sessions. Golden era of microbiology is 1857 to 1914. It is beginning of Pasteur's work. Discoveries include the relationship between microbes and diseases, immunity and antimicrobial drugs. The germ theory of disease given by Joseph Lister use a chemical disinfectant to prevent surgeries wound infections after looking at Pasteur's work showing microbes are in the air can spoilage fruit and cause animal diseases. In 1876 by Robert Koch provides proof that bacteria cause anthrax, provided the experimental steps Koch postulates used to prove that a specific microbes cause a specific disease. Koch's postulate says the specific causative agent must be found in every case of disease The disease organism must be isolated from the lesions of the infected case and maintained in pure culture. The pure culture inoculated into susceptible or experimental animals should be produce the symptoms of the disease. The same bacterium should be re-isolated in pure culture from the inoculated infected animal. Fermentation and pasteurization. Pasteur showed that microbes are responsible for fermentation. Fermentation is the conversion of sugar to alcohol to make beer and wine. Microbial growth is also responsible for spoilage of food. Bacteria that use alcohol and produce acetic acid spoil wine by turning it to vinegar. Pasteur demonstrate that these spoilage bacteria could be killed by heating that was not hot enough to evaporate the alcohol in wine. This application of a high heat for the short time is called pasteurization. This list shows the milestone and renowned microbiologists who got Nobel Prize in respective years for the correlated areas. Medical microbiology is the study of microbes that cause disease in human. Microbes are everywhere within the outside or inside of our body. Most of bacteria are harmless. Type of microbes include virus, bacteria, archaea, fungi, protozoa and microscopic algae. This slide shows protozoa, alga, bacteria. Diversity of microbes include bacteria, single celled prokaryotes, protozoa eukaryote single celled, fungi absorb nutrient, single celled filament, virus acellular entities and other worms and insects. This picture shows prokaryotic bacteria. In this picture we can see the chromatin material loosely scattered in the cytoplasm. This means it has absent of defined nucleus and hair like projections seen in the surface called pili and long hair like projections seen outside the bacteria is flagella. Flagella helps bacteria to move from here and there towards the nutrients and away from the toxins, and pili helps bacteria to attach from the surface. This picture shows eukaryotic cell. We can see well defined organelles, membrane bound organelles like nucleus, mitochondria, Golgi complex, which are usually absent in prokaryote like bacteria. Bacteria are the organisms reproduce asexually by binary transverse fission. They do not possess the nucleus typically like eukaryotes. The cell wall of these organisms are rigid, which important for the protection from external atmosphere, and mycoplasma, which is cell wall deficient organism, and rickettsia, which are absolute intracellular organism. Rickettsia, these organisms are obligate intracellular parasite, rod shaped to cocoid, and mycoplasma, these are bacteria which has deficient rigid cell wall. Two kingdom of bacteria, eubacteria and archaebacteria. Eubacteria are true bacteria and archaebacteria are oldest organism on the earth living in extreme conditions. This picture shows the gram positive bacteria. Gram positive bacteria retains the stain of crystal violet during gram stain. It has abundant amount of peptidoglycan. Gram negative bacteria, it sees pink under microscope. This bacteria has very thin layer of peptidoglycan. These bacteria cannot retain the gram stain and it takes counter stain from safranin. Antibiotics work by preventing cell wall formation, breaking up cell membrane or disrupting chemical processes. Antibiotics cannot treat viral infections. Fungi are eukaryotes, chitin walls present, use organic chemicals for the production of energy. Molds and mushrooms are multicellular, consisting the means of mycelia, which are composed of filaments called hyphae. Yeast are unicellular. Protozoa are eukaryotes, absorb or ingest organic chemicals for the production of energy, may be motile via pseudopodia, cilia or flagella. Most are free from parasites. Alga are eukaryotes. Alga contains cellulose. Alga contains chloroplast, so that they can utilize sun energy for the production of food or energy. These molecules produce oxygen and organic compounds. Metabolically, we can see metabolically diverse alga. Viruses are acellular organism consisting DNA or RNA in its core. Core is surrounded by a protein coat. Coat may be enclosed in a lipid envelope. Viruses are replicated only when they are in a living host cell, means virus cannot replicate itself, they cannot survive without any host cell. Multicellular animal parasites are eukaryotic type of organism. Parasite includes tapeworm, fishworm, roundworm. They are helminthic in type. They are microscopic in its stage of life. Bacteriology, study of bacteria known as bacteriology. It is the branch of speciality of biology that studies the morphology, ecology, genetics and biochemistry of bacteria, as well as other aspects related to them. The study of harmful effect of bacteria known as medical microbiology. In 1683, Antoine Leeuwenhoek describes bacteria as streak and threads under microscope, and in 1773, Danish scientist Friedrich Muller describes bacteria as bacilli, and bacteria word is coined in 1850 by French investigator. We can see the simple structure of bacteria. We can see a small hair like projections outside the surface of organism is pili, and one long thread like structure, it is helps for the movement of bacteria, is called flagella. In the cell, we have seen outer capsule, it is protective covering for the bacteria made up of polysaccharide, is protects from phagocytosis. Cell envelope includes 2 to 3 layers. The innermost layer is cytoplasmic membrane. It envelops the cytoplasm, which contains nuclear material, ribosomes, but it is absent a defined nucleus and membrane bound organelles. The outer layer of cell membrane is cell wall. Bacterial cell wall includes peptidoglycan in both gram positive and gram negative, but it is thick layer in gram positive, whereas thin layer in gram negative. Cell membrane envelops the cytoplasmic membrane. It is made up of phospholipid and proteins. It always allow molecule to move in and out. Cytoplasm helps bacteria to grow and metabolism and replication. Flagella is a long hair like structure. It is like a propeller. It helps to move bacteria towards the nutrition and away from the toxin chemicals. Pili are the small hair like projections emerges from the outer side of cell wall. It helps bacteria to attach on the surface. Nucleoid is the bacterial chromatin, because bacteria has no defined nucleus. So, nuclear material is scattered in the cytoplasm, so known as nucleoid. Mesosome is the inner folding of bacterial cell membrane. It is similarly the mitochondria in eukaryotes. Plasmid is the circular small double helical DNA. It is distinct from the cellular chromatin. Plasmid is naturally exist in bacterial cell. Plasmid is targeted for the genetic engineering and molecular biology. Nowadays, plasmid gives additional advantage to the bacteria like antibiotic resistance. Ribosomes are present in both prokaryotes and eukaryotes. In prokaryotes, it is 70S type, subunits are 30S and 50S, whereas eukaryotic ribosomes are 80S type, subunits are 40S and 60S. We can classify bacteria in various ways like according to the shape of bacteria, according to the cell wall of bacteria, according to the flagella present on the bacteria, according to the nutritional mode of bacteria, according to the temperature where bacteria can survive. On the basis of shape, we can classify bacteria in various ways like cocci, bacilli, vibrio and spirilla. Cocci are spherical in shape. We can further classify cocci in coccus, streptococcus, diplococcus, staphylococcus, tetrad and Yersinia according to the cocci arranged in groups. This slide also shows the same thing. One cellular coccus is coccus, 2 cell arrangement is diplococcus, 4 cell arrangement is tetrad, 8 cell arrangement is Yersinia, straight chain arrangement is streptococcus and grape like arrangement in group is called as staphylococcus. This picture also shows the streptococcus and staphylococcus and also seen the diseases which can caused by this bacterial pathogens. Bacillus is rod shaped cell, it is gram positive in nature, it is obligate anaerobe, but facultative anaerobes also. Obligate aerobe means require oxygen for the survivalship and facultative anaerobe can survive with or without oxygen. Bacillus can classify according to the number of cells arrangement. One bacteria present as a bacillus. If bacteria is transverse oval is called Coccobacilli, 2 cell arrangement is Diplobacilli, straight chain arrangement is Streptobacillus and palisade if irregularly is chain present. Vibrios are comma shaped cells. It is gram negative in nature. It is causative organism for most common foodborne infections. It survives in high salt medium, but cannot survive in fresh water. Spirilla is helical shaped bacteria. It is characterized third most common morphology after bacilli and cocci. Spiral bacteria can be classified by the number of twists per cell, cell thickness, cell flexibility and motility. Two types of spirilla we can see, one is spirillum, second is spirochete. Spirillum is rigid external flagella and spirochete is flexible internal flagella. According to the gram staining, bacteria can be classified as gram positive and gram negative. Gram positive bacteria has thick layer of peptidoglycan, whereas gram negative has very thin layer of peptidoglycan. Gram positive bacteria has teichoic acid, whereas absent in gram negative. Gram positive bacteria is so called because it retains gram stain and appears violet under microscope, whereas gram negative bacteria cannot retain gram stain and appears pink under microscope due to safranin as counter stain. This is the process of gram stain. Firstly, we stain bacterial smear by crystal violet and iodine, and after staining we use alcohol as a destain and counter stain as safranin. This is the process of gram staining. We have discussed earlier, gram stain is preferred on a smear prepared that has been fixed. One function of fixation is to secure the cells in the slide. We can classify bacteria on the basis of nutrition, photoautotroph, photoheterotroph, chemoautotroph and chemoheterotroph. We can also classify bacteria according to the flagella present on the bacteria. Atrichus has no flagella on the bacteria, Monotrichus one flagella on the bacteria, Lophotrichus tuft of flagella and one end of bacteria, Amphitrichus tuft of flagella present on both end of bacteria, Peritrichus flagella present all over the bacterial cell. In this slide, we can see the flagellar arrangement which we discussed earlier. Bacterial reproduction by two mode, asexual and sexual, asexual by binary fission mainly and sexual reproduction by conjugation, transformation and transduction. Asexual mode is most common mode. Bacteria primarily use binary fission as the asexual reproduction mode. Endospore formation is the mode of reproduction in which bacteria form endospore for the extreme atmosphere, in which bacterial cell is concentrated around the nuclear chromatin and hard capsule secreted around the cell. Vegetative reproduction of bacteria by binary fission, budding and fragmentation. Binary fission is most common mode. In this mode, firstly bacterial DNA replicate and form a duplicate copy of DNA. DNA moves towards the pole and from the center a notch is formed which divides the cell into two. By budding, in this type of reproduction, firstly a bud is formed which is gradually bigger in size, nuclear chromatin is replicated and one part is enter in the bud and bud is separated from the main cell. In fragmentation, firstly nuclear chromatin is replicated many times and cell is fragmented into many parts and each part is known as gonadia. This type of reproduction we can see in the unfavorable condition. When favorable conditions come, this gonadia can generate a new bacterial cell. Sexual reproduction, it is conjugation, transformation and transduction type. Conjugation is the most common mode of sexual reproduction. The donor cell marked as X plus F plus and recipient cell is marked as F minus. Physical contact between donor and recipient cell. The pilus form the conjugation tube, enables direct contact between the donor and recipient cell. Transfer of F plasmid cell from F plus to F minus. Transformation. Bacterial transformation is a process of horizontal gene transfer by which bacteria takes a foreign DNA material. Transformation does not require living donor cells. Once the transforming DNA enters the cytoplasm, the exogenous genetic material is similar to bacterial DNA. It may integrate into the chromosome and it is important for the genetic engineering. Transduction is the mode of genetic transfer from one bacteria to another by a virus. Once bacterial cell attacked by a virus entered in the bacteria and genetic material integrate with the host cell and multiply. Once multiply viral genome, it separate from the host cell and again packing of virus, and after burst of host cell virus released. In this packing, some virus cells also takes host cell which transfer the other bacteria when it attacks to the next generation of bacteria. This picture shows the steps of transduction. Growth curve includes generation time, the amount required for the doubling the population. Growth curve has 4 phases: lag phase, exponential phase, stationary phase and decline phase. In lag phase, the additional period which requires the bacteria adjusting to the new conditions. Once bacterial cell is accumulated all the needs of growth and process for the cell division, the exponential or log phase of growth is marked by predictable doubling of the population, where one cell become 2 cell, become 4 cell and become 8 cell in further sequential division. In optimal condition, very rapid growth, while less ideal condition will slower the growth. Due to the probability of growth in this phase can be used to mathematically calculation with the time by this formula, in which G is generation time represented by T by N. T is time, N is number of cycle. Stationary phase, at some point of bacterial population run out of an essential nutrients and chemicals, growth is inhibited by its own waste products or lack of physical space. Death or decline phase, the last phase of growth curve, the number of viable cell decreases in a predictable fashion. The steepness of the slope corresponding to the how fast cells are losing viability. Culture conditions have deteriorated to a point where the cell are irreversibly harmed, since cells collected from this phase fail to show growth on fresh medium. Measure of growth of microbials by number of cells or by change in mass. Direct cell count and viable cell count are the mode of measurement of growth. In direct cell count, counting chamber, electronic chamber on membrane filtration. Visible cell count is plating and membrane filter method. Beneficial effect of bacteria seen in dairy product, pickle industries, importance in sewage treatment, antibiotics and gut flora. Harmful effects we can see in the form of various diseases like tuberculosis, pertussis, diphtheria, tetanus, toxoid, meningitis, gonorrhea, pneumonia and syphilis, etc. Thank you.